Hey everyone, welcome back. Today we are unpacking a seriously strategic blueprint in this explainer. We're going to look at the incredibly meticulous process of architecting a completely fail-proof cell therapy and how doing that means integrating cGMP right from the very start. Let's translate some brilliant industry insights into a crystal clear path to actual commercial viability.
Think about this for a second. What if delaying regulatory compliance is the exact reason a cell therapy fails? I mean, it's a massive misconception that cGMP is just some late-stage regulatory burden or, a box you check at the end of the line. Treating it that way is exactly how groundbreaking therapies run out of capital and literally never reach the patient.
Building a viable cell therapy is a lot like architecting a highly advanced, structurally sound building. You can't just slap a foundation under a skyscraper after it's already built, right? The blueprint for your facility, your technology, and your compliance has to be integrated from day one. If your structural architecture isn't sound, the science simply cannot scale.
All right, so here's our blueprint for today. First, the imperative and complexity. Second, the 4 cGMP pillars. Third, the QA checkpoint lifecycle. And finally, verification versus validation. Let's get right into it. Part 1. The imperative and complexity of cell therapy. Because, every solid building needs a strong foundation, and ours starts with understanding biology and timing.
Take a look at this cost-time curve. It is a harsh reality check. Early cGMP integration definitely requires substantial upfront capital. But if you adopt it late, the cumulative risks and costs absolutely skyrocket. You get hit with massive financial penalties, tech transfer bottlenecks, retrospective engineering, and worst of all, outright regulatory rejections when you finally try to get your IND or NDA licenses.
So why does that huge spike happen? It all comes down to the biology. The financial risks we just mapped out are a direct result of biological complexity. We aren't just mixing powders in a beaker here, we are working with living materials. The later you try to force rigid controls onto unpredictable biology, the more incredibly expensive and chaotic it becomes.
Think about standard chemical molecules. They're static, highly uniform, and strictly controlled by a basic chemical formula. But living cell therapies, they have off-the-charts biological variability. Because the inputs are just inherently unpredictable, the manufacturing process itself literally has to become your ultimate quality control mechanism.
And this brings us to the crucial bottleneck, what we call the scale-up chasm. Crossing from early scientific feasibility in a GLP lab over to the hardcore commercial rigor of GMP is a massive, massive leap. Most cell therapies fail right here, simply because they aren't structurally ready for FDA or EMA scrutiny.
Moving on to Section 2, the 4 cGMP pillars. Time to actually erect the operational pillars that build this solution.
Look, regulatory compliance isn't just a to-do list. It is a fully integrated system. The whole framework revolves around patient safety, product integrity, and continuous improvement, and it's all held up by 4 completely non-negotiable pillars: hardware, technology, documentation, and personnel.
Let's look at the first pillar, hardware. The physical environment has to be ruthlessly controlled. We aren't just talking about basic clean rooms. It's about deep layers of defense. Imagine you build a state-of-the-art facility on top of a mountain. A single insect breaching the perimeter of your Grade A zone causes a total, absolute operational shutdown. Your physical location and your HVAC redundancies are literally active components of your manufacturing process.
And here I really need to emphasize the absolute golden rule of cGMP. If it is not documented, it did not happen. Period. Without bulletproof, rigorous data capture, that billion-dollar physical mountain fortress we just talked about, it essentially means nothing to a regulator.
Our next pillar is technology. Think of this as the translation engine. This is where you grab raw scientific concepts from an academic setting and aggressively refactor them into highly standardized, validated GMP protocols. Your analytical methods for identity and potency have to be locked down tight to prove you can safely and consistently reproduce the efficacy of a living product.
Next up is personnel. Now, in an early startup lab, you've got cross-functional researchers doing a little bit of everything. But at the cGMP level, having operational overlap like that is a massive risk. Every single step requires highly specialized, dedicated personnel, and they have to be overseen by a completely independent quality assurance unit.
Our final pillar is documentation. Think of this as the core operating system of your facility. From master batch records to SOPs for personnel management, facility rules, material sourcing, these 6 codes form the legal and operational backbone that integrates and runs the entire system flawlessly.
Within that documentation, traceability is quite literally life or death. GMP mandates relentless data capture. We're talking 10-year continuous temperature storage logs and closed-loop barcode tracking. In autologous therapies, where you take blood from patient A, process it, and infuse it back into patient A, a mix-up is fatal. Flawless traceability is your ultimate liability protection.
All right, Section 3, the QA checkpoint lifecycle. Because frankly, our architecture is only as robust as the security checkpoints actively guarding it.
Here's the thing. Quality assurance is not just some final inspection at the very end of the assembly line. It is an active, embedded toll gate throughout the entire 11-stage product lifecycle. If your process fails at any single toll gate, you don't just, tweak it and move on. It forces a complete and incredibly costly process reset.
Now, to simplify that kind of overwhelming 11-step framework, let's just zoom in on 3 of the absolute most critical, non-negotiable checkpoints where therapies are most frequently halted. Step 1, donor screening, which involves rigorous third-party viral checks. Step 2, formulation, ensuring exact drug substance specifications. And Step 3, final product QC, matching all analytical documents perfectly before product release.
Which brings us to our final part, Section 4, verification versus market validation. Because even the most flawless physical building is totally useless if it's built for the wrong purpose.
Think about the real difference between verification and validation. Verification asks, did we build the product right? But validation asks, did we build the right product? Imagine engineering a flawless blood pressure monitor with huge, highly functional buttons. You verified it works perfectly. But if you never validated that the senior citizen market actually wants or needs that specific monitor, well, you've just engineered a commercial failure.
So, what you really should take away from this is that strategy dictates architecture. You have to work backwards. The smartest biotech companies start at Step 1. They define the clinical market reality and validate the demand first. Then they define the target clinical efficacy. Only then do they custom engineer their cGMP process backwards to deliver those exact verified specifications.
To sum this up with the gravity it deserves, early cGMP adoption is not a regulatory penalty. Actually, scratch that. It's the exact opposite. It is the absolute foundational architecture that guarantees product safety, protects investor capital, and acts as the true engine securing your market dominance.
I want to leave you with a final, provocative thought, something to help you critically reassess your own timelines. Are you right now architecting a perfect, verified facility for a cell therapy that the market doesn't even want? Take a really hard look at your blueprint, validate your market first, and integrate cGMP from day one. Thank you so much for joining me in this explainer, and keep building brilliant things.